I did my summer project with Dynaval, and the title of my project was the impact of ambient exposure as a consequence of mishandling on bull spermatozoa motility. So just a little bit of background. So artificial insemination is an invaluable tool to the agricultural industry, notably the dairy industry. And it's largely facilitated by cryopreservation, which allows long-term storage of superior bull semen samples. So these bull semen samples are typically cryopreserved within the 0.25 ml or 0.5 ml cryo straws, which are shown in the image here. And they're maintained within these goblets that are submerged in liquid nitrogen at minus 196 degrees in these dur canisters in the bottom figure. So when a straw is going to be used for insemination, it's thawed approximately 35 to 37 degrees. But prior to this, there's multiple points where ambient exposure could occur, resulting in prematurely breaking the cold chain and significant temperature fluctuations, which could have negative consequences on the semen biology and therefore fertilization capacity. So the first stage where this ambient exposure would typically occur is as a single ambient exposure. So in the straw redistribution stage at AI commercial sites, straws will often be repackaged into different ordered quantities to be sent off to the relevant farmers. At this point, straws may be temporarily moved between different tanks for transport or long-term storage. And although these ambient exposures are brief, they could be significant. And the second stage where ambient exposure occurs is as a multiple ambient exposure, which happens when the AI technician is performing the insemination itself. So as I said, these straws are kept within the goblet that are shown in the figure. And when our AI technician is going to inseminate a cow, they'll lift the goblet to the neck of the canister, or perhaps even in some really poor handling practices, somewhat outside of the canister, select the correct straw for insemination, and then return the goblet back into the canister, which results in the remaining straws, which can experience multiple thermal fluctuations over a long time course before they're eventually thawed for use. So the aim of my project was to really look at this and to assess the impact of these ambient exposures which do occur due to these poor handling practices and how this might affect motility parameters. The hypothesis for my project was as you increase the duration and number of ambient exposures, motility parameters that I'll talk about in a minute will decrease. So these are my methods that I performed with regards to the laboratory work. And as I previously talked about, you get single ambient exposures and multiple ambient exposures, and my experiments were meant to mimic these sort of poor handling practices. So the figure here on the left, the single ambient one for stage 1 shows the straw's usual position within the tank. It's kept within one of these goblets that hangs inside of a dur canister. For the control straw for both experiments, the straw was taken out with tweezers and immediately thawed in a water bath. After 40 seconds in a water bath, the straw was removed, cut and emptied into a centrifuge tube. And then 3 microliters of the sample was micropipetted into a ledger slide, which is a fixed depth chamber slide. This ledger slide was then inserted into the Dynascan for analysis, shown in step 5, which provides measurements on the mean swimming speed of a sample and the percentage progressive motility, which is a percentage of spermatozoa which display rapid linear movement, which is required for successful fertilization. The test straws for these experiments are shown by 2b. They were held outside of the dur canister with the tweezers for either 5, 10, 20, 30 or 60 seconds to get the full sort of time scale of the duration of ambient exposure, and then refrozen in the tank for 15 seconds before being thawed in the water bath and following the same steps for Dynascan analysis. Then for the multiple ambient exposures, this shows the straw's usual position within the tank again in step 1, and we then did a pilot study where the straw was held in the goblet outside of the dur canister shown in 2a for first just a single cycle of 1 minute or 2 minutes. And then after this, we looked at increasing this number of ambient exposures. So we increased this to 10 cycles of 1 minute, every time being held outside for 1 minute, and then being refrozen in the original position within the tank, shown in number 3. And then did this for 30 cycles as well to look at the impact of increasing this cycle time. Then to compare this with another poor handling practice, which is holding the straw in the neck of the canister because elevated temperatures are still present here. The straw was held within the goblet, which was raised within the canister again for 10 or 30 cycles, refrozen in between each cycle. And then all of these test straws basically followed from step 3 for the single ambient exposures, where they're thawed, prepared, and then inserted into the Dynascan for analysis. So this here is the first result that I got. These are all averaged over replicates. So it was for the single ambient exposure that I was talking about. And in figure A, this is for the progressive motility. So what was very clear was there was a significant negative correlation between percentage progressive motility and ambient exposure. And as you increase the duration of ambient exposure, there was almost a linear relationship with progressive motility going down. Even a 10-second single ambient exposure, which is obviously pretty brief, still had an 11.71% mean reduction from the control progressive motility. Whereas when you compare this with the swimming speed, the swimming speed wasn't really as impacted between the conditions. Most mean swimming speed values kind of ranged from 57 to 65 microns per second. And then when I did statistical analysis, it indicated that the progressive motility values were significant from one another in an ANOVA, but the swimming speeds values were not. And then the second result. So this is for the multiple ambient exposure experiments I talked about. And first for figure A, these are the cycles that are done holding the straw in the goblet outside of the neck for 10 or 30 cycles, or inside of the neck for 10 or 30 cycles. And both of these are for progressive motility. So it's not, the results from the pilot study aren't shown here, but they basically resulted in a non-significant reduction from the control. However, when you increase the single 1 minute ambient exposure to 10 cycles of 1 minute ambient exposure, for figure A, there was almost a twofold reduction in progressive motility. So it was indicative that something is a single cycle, which may not induce sort of damage to motility, as multiple cycles could induce very significant damage. And then as this was increased to 30 cycles, this wasn't statistically significant from the 10 cycles, but still significantly reduced from the control. Then from figure B, the exposures which were inside of the neck of the canister showed a slight reduction across the number of cycles, but this wasn't actually statistically significant, which is hypothesized to be due to the level of liquid nitrogen not varying as much, whereas when you do these ambient exposures outside the neck, the level of liquid nitrogen rapidly evaporates and obviously results in a lower temperature. And the swimming speed for the multiple ambient exposure experiment. Again, the swimming speed doesn't really seem to be impacted by these ambient exposures. It kind of sits between 60 to 65 microns per second, and it's not statistically significant between test groups. So this is so for what these results really meant, I guess the project really did show that as you increase the duration of ambient exposures, and also as you do increase the number of cycles, it can have very significant impacts on the motility and therefore fertilization capacity of these bull spermatozoa. What was clear between different ambient exposures, however, was that the parameter that was always impacted was the progressive motility and not really the speed, which indicates that this with ambient exposure, the percentage of cells which displays rapid linear movement are impacted between different conditions, but they move at a sort of consistent speed to the control population. In terms of previous literature, which has done similar things, it is actually quite limited. But from single ambient exposures, a lot of papers recommend that the maximum moving time between tanks is 3 seconds, which indicates that any time above this may compromise the quality of the straw. For multiple ambient exposures, we demonstrated that when you increased from a single 1 minute cycle to a 10 cycles outside of the neck of the canister, this did significantly decrease progressive motility, whereas when you did this in the neck of the canister, up to 30 cycles still didn't show a significant reduction from the control. And these neck ambient exposures in the canister match the results of a previous paper by Lieberman et al., who demonstrated that 20 one-minute ambient exposures within the neck of the canister don't significantly impact motility parameters, but they do, interestingly, significantly impact acrosome integrity, which is maybe something that could be looked at in a further study. Another point for further study would be to increase the cycle number within the neck of the canister to an even higher cycle number, because Pace and Sullivan demonstrated significant damages to motility after 480 cycles over the course of 1 year that was conducted on a farm. So the proposed reasons for behind these damages that occur with ambient exposure is due to cold shock and ice crystal formation. Because of ambient exposure, you're allowing the temperature of the straw to rise within perhaps the danger zone hypothesized for ice crystal formation, which is 0 to minus 50 degrees. There is a lot of scope for further research. So in terms of looking at why we do see this reduced motility with ambient exposure, we could look at membrane viability to see if the cells that are motile are actually dead, and we can't just infer that they're metabolically inactive. So you could do that using dye exclusion tests with fluorescent microscopy, or using the CASA, the computer-assisted semen analyzer. Or we could also look at mitochondrial integrity and structure, because if there's ice crystal damage to the mitochondria, it may be inhibiting ATP metabolism and therefore motility. And that could be done doing looking at transmission electron microscopy for the structure. So this is the impact, the overall impact of my project, which I guess is aimed more at perhaps non-scientists. So the statement is poor fertility costs, and the reason why I chose the statement really links back to my introduction slide. So poor fertility, especially male infertility due to poor handling practices, does have a large economic sort of burden on the dairy industry, in terms of if cows aren't getting pregnant, it reduces calving sales and dairy sales, but it also has a large environmental cost. So if cows aren't getting pregnant as a result of poor semen quality, then quite often there's a higher number of cows that will be culled, a higher number of replacement cows that are required to sustain fertility within that population, and then more methane emissions, which obviously has a negative impact on the planet in terms of greenhouse gases. Thank you for listening, and I'd be happy to answer any questions.